Welcome to another lecture in our Amino Acid Disorders series. Today's focus is on Fatty Acid Oxidation Disorders, or FAODs, a group of autosomal recessive metabolic conditions caused by mutations in genes encoding mitochondrial enzymes critical for beta oxidation of fatty acids. Key genes involved include AKDM in medium-chain acyl-CoA dehydrogenase deficiency, MCAD, ACADVIL in very long-chain acyl-CoA dehydrogenase deficiency, VLCAD, and HADA or HADHB in long-chain 3-hydroxyacyl-CoA dehydrogenase deficiency, LCHAD. These enzymes facilitate the stepwise breakdown of fatty acids to generate acetyl-CoA, FADH2, and NADH, essential for ATP production during fasting or stress. When these enzymes are defective, fatty acid intermediates accumulate, and energy production from fat becomes impaired. This results in hypoketotic hypoglycemia, hepatomegaly, cardiomyopathy, and rhabdomyolysis, particularly during periods of catabolic stress. Clinical presentation often begins in infancy or early childhood, with lethargy, vomiting, seizures, or sudden death in severe cases. Long-chain defects may also present with arrhythmias or skeletal myopathy. Diagnosis is achieved through newborn screening via tandem mass spectrometry, which detects abnormal acyl-carnitine profiles. Confirmatory testing includes targeted gene sequencing or enzyme assays. Management involves strict avoidance of fasting, use of high-carbohydrate diets, and in some cases supplementation with medium-chain triglycerides or triheptanoin. For patients with carnitine transport defects, carnitine replacement is required. Acute episodes are treated with intravenous glucose to prevent metabolic decompensation. When detected early and managed correctly, many patients lead stable lives, though the risk of metabolic crises persists. Subscribe for upcoming lectures as we continue exploring metabolic disorders and their systemic impact.